Good morning, students. Last class we have discussed about in asexual method of reproduction and its types. Last class we have detailedly discussed about in asexual method of reproduction in fission. Now we have discussed about in other types of asexual method of reproduction: budding, sporulation, fragmentation, gemmal formation, and regeneration. First we started in budding. Budding is the process where the parent body produces a one or more buds and each will grow into young one. A parental body produces a bud-like structure and each bud is furtherly developed and to form a new individual. Example, hydra and sponges. The buds is classified into two types depending upon their growth. So namely exogenous buds and endogenous buds. First one, exogenous buds. The buds developed from outer surface of the body. So outer structures of the body. So see the pictures. Hydra, it's form a buds or initiate the bud-like structures. Furtherly, buds is developed. The completely developed buds is disattached from the parental body. It is examples of exogenous buds. So it is a microscopic image of budding process of hydra. Next one, endogenous bud. The buds developed inside the cytoplasm and remain within the parent body. Buds developed and remaining there inside of the parental body. Such kinds of buds is called endogenous buds. Examples, Nactiluca. Next types of asexual method of reproduction, gemmal formation. So gemmules are the special types of internal buds produced by a member of Spongilla family sponges. During an adverse condition, adverse means condition is unfavorable. During the adverse conditions is adverse or unfavorable, the gemma is developed. A gemmules is a hard ball-like structure having a mass of food-laden archaeocyst. So gemmules, just like a hard balls, it contains a food-laden archaeocytes. Archaeocytes means initiated cells. A gemmules can withstand adverse conditions and they begins to hatch on the return of favorable conditions. A gemmules withstands adverse conditions. Conditions when favorable, the gemmules ruptures and each post is developed into a new individual. See the pictures. It is a gemmules or hard ball-like structure contain a food-laden substance of archaeocytes. It is a gemmules. One enlargement structures is the transfer structures of gemmule. Next types of asexual method of reproduction, fragmentation. Fragmentation is a types of asexual reproduction in which the parent body breaks into fragment and each fragment has the ability to develop into a new individual organism. So parental body splitting or dividing or broken in a many pieces. So each piece developed into a new individual is called fragmentation. So fragmentations in sea animals is referred as a petal lacerations. When sea animals gets disattached and move, the petal disc break up. So lobes are constructed from a leaf-like structures and each lobes developed into an entire organism. See the pictures, sea anemone. It is a petal disc. So it's a leaf-like structure. This leaf-like structures is considered to be a lobes. Then each lobes is developed into a new individual. Next one, fragmentation in tapeworm. Tapeworm, this is a tapeworm, so mature tapeworm structures. So already you know in previous class, phylum Platyhelminthes. So tapeworm is a ectoparasite in human being. It depending upon the two types of host complete in life cycle. So fragments of the tapeworm. In tapeworm, the mature proglottists at the posterior part of the body get periodically cut off. This shedding of gravid proglottists by tapeworms is called apolysis. Apolysis is a very important question. So what is meant by apolysis? The mature proglottist of the tapeworm is disattached from the parental body. This proglottist contains the embryos and then entering the host, developing into a new individual. This proglottist or mature proglottist disattached from the parental body. This proglottist contain a embryos. It is a mature proglottist. It's also a mature proglottist disattached from the parental body. It contains a mature embryo. So it's entered to the secondary host in pig, developed into a new individual. It's depending upon them two types of host complete the life cycle. The primary host is a human being or man. The secondary host is a pig. The apolysis is significantly helpful to the transfer of the matured embryo from one host to another. Next types of asexual method of reproduction is regeneration. Growth of the injured region is called as regeneration. Regeneration was first studied in Hydra by Abraham Template in 1740. The regeneration is two types, morphallaxis and epimorphosis. See the pictures. Tail losing lizards. So ladrons, the loosening part or losing part of the tails is completely developed. It is a regenerations. First types of regenerations is morphallaxis. Morphallaxis refers to the regenerations of the entire body from the cut off fragment. A small piece of the organism able to produce a entire or completely developed one. A small piece of the organism. It is a piece of organism. It is a piece of organism. It is a piece of organism. This piece of organisms is able to produce a losing part of the other body to develop a complete whole organism. So regeneration in planaria. This is a regeneration in hydra. The hydra is cut at a two end, anterior and posterior. The anterior ends is furtherly developed into produce a entire complete hydra. Then posterior end is continuously developed and finally form a entire hydra. It is a morphallaxis. Next one, epimorphosis. This is second types of regeneration. Epimorphosis is refers to a replacements of the body parts. Body part replacements is also known as epimorphosis. So epimorphosis is again classified into two types, reparative regeneration and restorative regeneration. Only certain damaged tissues are generated or regenerated is called reparative regenerated. Examples, regenerations of a damaged liver tissue. For examples, the liver is infected or damaged in a various regions. The damaged liver tissues is replaced or regenerated. This process is also called as epimorphosis. Next one, restorative regeneration. The served body part are regenerate. Example, regenerations of the lost tails of a lizard and arms of starfish. Starfish or sea stars losing the one arms, then later on it's developed the loosening part and form a complete whole organism. This is the epimorphosis. Important questions for the difference between morphallaxis and epimorphosis. So now we are completed in asexual method of reproductions. So last class we will see about the asexual method of reproduction and its types of asexual method of reproduction in fission, then sporulation, budding, fragmentation, multiple fission, gemmel formation and regeneration. Then next we see the next types of reproduction in sexual reproduction. Sexual reproductions involve the complete fusions of male and female gametes to form a diploid zygote which developed into a new individual. So asexual method of reproduction does not form any gametes and does not gamete fusion, but a sexual method of reproduction, so gametes is developed and formed, fused to form a diploid zygote which develops into a new individual. Sexual reproductions lead to genetic variation due to recombination of genes, but asexual method of reproduction does not lead to any variation because it's involving a single genetic parent. Sexual method of reproduction is classified into two types, namely syngamy or fertilization. Next one, conjugation. Note the important questions. Write any two difference between syngamy and fertilization. So syngamy are also known as fertilization. It is a diagrammatic representations of a sexual method of reproduction. Fusions of the two different types of gametes. This is a male gametes, this is a female gametes. Both are fused and form a diploid zygote. A zygote furtherly undergo a cell division, form a embryo. So embryo developed into a young one. So types of fertilization. First types of sexual method of reproduction, fertilization. So fertilization is classified into two types based on the site of occurrence. So fertilization takes place, it's based on two types, external fertilization and internal fertilization. So fertilization, external and internal. First one, external fertilization. The fusions of male and female gametes occurs the outside of the body and organism. Fertilization takes place in outside of the body is called external fertilization. Fertilization takes place in inside of the body is called internal fertilization. So major difference, fertilization takes place in outside of the body is called external fertilization. Fertilization takes place in inside of the body is called internal fertilization. So it is a examples of external fertilization fishes. So see the picture. It is a sperm. So releases in the male fish for water. So it is here eggs. It releases from female fishes in water. Both sperms and eggs are fused. Both sperms and eggs are fused to form a diploid zygote. Furtherly, the zygote developed into a new individual. It is examples of external fertilization. Next one is examples of internal fertilizations. Reptiles, amphibians, mammals. Say examples of internal fertilization. Next, types of syngamy based on the morphology and production of gametes. So syngamy is classified into few types based on the morphological and the production of gametes. First one, autogamy, then exogamy, halogamy, then pedogamy, merogamy, isogamy and anisogamy. So these are the types of syngamy based on the morphology and production of gametes. First one, autogamy. Male and female gametes are produced by a same or same body by gets fused to form a zygote. Male and female gametes. Both the gametes are developed in a single individual, and fusions of gametes to form a organisms is called autogamy. See the pictures. It is the examples of autogamy in paramecium cells. So contain two types of gametes, male and female. So macro and micronucleus. The nucleus are fused and developed in a zygotes furtherly divided. It is a examples of autogamy. Next one, exogamy. Male and female gametes are produced in a different body. Male and female gametes are developed into a different body and they both get fused to form zygote. So, both male and female gametes developed into a different organisms and fused together form a single zygote is called exogamy. Next one, hologamy. So no gametes are formed. Instance, the entire matured organisms behave like a gamete and fuse with the other mature organisms. Hologamy, the organisms does not form any gametes, but the entire organism act as a gamete. Okay, hologamy means the organism does not form any gametes, but the entire organisms, it is a organism, it does not form any gametes. The entire organism act as a gametes. The mature organism fused with another one mature organism. Both the organism are act as a gamete. These types of gamete fusions or syngamy is called halogamy. Next one, pedogamy. Sexual union of young individual produced immediately after the divisions of adult parent cell by mitosis. Pedogamy means sexual unions of young individual produced immediately after the division of adult parent cell by mitosis. So see the picture. It is a divisions of nuclei. A single cells divide into two nucleus initially, then later on two separate nuclei is formed, or single cells contain two different nucleus. Single cells contain two different nucleus. Then each nucleus is immediately fused to one another. Each nucleus is immediately fused to one another. The young nuclei is fusions, and young nuclei fuse to form a diploid zygote. Diploid zygote. This is also known as pedogamy. Sexual unions of young individuals. The cells contain two nucleus or newly formed two nucleus immediately fused to form a diploid zygote is called pedogamy. Next, merogamy, isogamy and anisogamy. So merogamy refers to the fusions of small sized and morphologically different gametes. The gametes are very smaller in size and morphologically different. It is called as merogametes. Merogamy. The gametes is smaller size. Gametes is very smaller size and morphologically different gametes is called merogametes. Next one, isogamy. Iso means similar or same. Isogamy is referred to the fusions of morphological and physiological similar gametes. Both morphological and physiological similar gametes is called isogamy. Next one, anisogamy. Anisogamy refer to the fusions of morphological and physiological dissimilar gametes. Both the morphological and physiological dissimilar or a different gametes is called as anisogamy. So write the difference between isogamy and anisogamy. Iso means same or similar. Aniso means dissimilar or different. The pictures, it is a isogamy. Both the gametes, one male and female, two gametes are similar, same structure or same morphology. So morphologically and physiologically similar gametes is called isogamy. Next one, anisogamy, morphologically and physiologically dissimilar. So it is a larger one gametes, it is a smaller one gametes. It is a larger one, it is a smaller ones. Both are different. Both are different. This types of gametes is called anisogamy. Next one, second types of sexual method of reproduction is a conjugation. So conjugation is the temporary union of two individuals of the same kinds. Same types of individuals temporarily union to exchange the genetic material from one another. During the conjugation, a small portions of a nuclear materials is transferred from donor organism to the recipient organism. Example, bacteria and paramecium. So already you know the bacteria and the paramecium are examples of asexual method of reproduction. So some of the bacterium and paramecium in conditions is unfavorable or favorable situations, it's lead to sexual method of reproductions, namely conjugations. So see the pictures. This is a bacterial conjugation. So one types of bacterium contain a plasmids and a mobile plasmids, then phyllis. So already you know about the structure of bacterium. The bacterium is surrounded by a capsule. The outer structures of the bacterium contain a small thread-like structures. A small fine thread-like structures is called villi. So it's help in locomotion of the organism. Some of the villi involving in sex linkage or transfer the genetic material, such villi is called sex villi. It is a sex phyllis. It helps to exchange the genetic material. It's help to exchange the genetic material. It is the sex villi. So it is a mobile plasmids. Plasmids. Already you know the term plasmid. Plasmid is a extra chromosomal DNA. So DNA only contained for a inside of the nucleus. Inside of the nucleus. If the DNA is found or presents, the other regions of the nucleus is called as extra chromosomal nucleus. The extra chromosomal nucleus or DNA is called an plasmid. So it is a donor, it is a recipient. The donor plasmids and recipient plasmids is contact with the help of phyllis or sex villi. The mobile plasmids is move or release from the donor to recipient through the sex villi or phyllis. The mobile plasmids is transferred from donor to the recipient. So after transformations of the donor to recipient, both the organisms is separate, and individual organisms are both donor and recipient to form a new daughters. It is a next types of sexual method of reproductions. Next one, phases of life. Phases of life. So lives having three phases: Johnlian phase and reproductive phase. Next one, senescence phase. First one's Johnlian phase. The period between the birth of an individual's organism up to its reproductive maturity. So newborn to until entered to the puberty is called Johnlian phase. The periods between them birth of an individual organisms up to its reproductive maturity, newborn to enter to puberty stages is called Johnlian phase. Next one, reproductive phase. So reproductive phase, the period where organisms have the ability to reproduce and gives rise to offsprings. The organisms able to reproduce and form a offsprings period is called reproductive phase. So classification of animals based on the breeding nature. The animals is classified based on the breeding nature. So seasonal breeding, organisms which reproduce only during a particular period of the year. Some types of organisms reproduce only for the particular year of the or particular season of the year. Such types of breeding is called seasonal breeding. So examples based on dogs. It's reproduced only in a particular period of the year. The next one, continuous breeders. Organisms which continues to reproduce throughout the sexual maturity. So organisms which continue to reproduce throughout the sexual maturity, throughout the year, so able to reproduce throughout the year. Such organisms or such method of breeds is called continuous breeds. Next phase, senescence phase. The period where the structure and functions of organisms gradually disintegrated leading to demise. So organisms where the structures and functions of the organism gradually disintegrate leads to the demise. So after reproductive phase 2, death is called senescence phase. Next one, parthenogenesis. It is a very important 5-mark question and 3-mark questions. Parthenogenesis. So see the picture. It is a completely stop or does not involved in a one types of gametes. It is a male. So male gametes does not involve to form a new individual. So parthenogenesis. Development of an embryo from an unfertilized eggs. Unfertilized eggs to form a new individuals is called parthenogenesis. It is the egg structure of eggs does not fusions of gametes. It's completely developed into larva, and the larva form a young one. This process is called parthenogenesis. So types of parthenogenesis: natural parthenogenesis and artificial parthenogenesis. First one, natural parthenogenesis. Parthenogenesis that occurs naturally in the life of an organisms. So parthenogenesis, it's naturally occurs in the life of the organisms is called natural parthenogenesis. Parthenogenesis means the eggs are young one to produce without fertilization. Without fertilization. So types of natural parthenogenesis: complete parthenogenesis, incomplete parthenogenesis, and pedogenetic parthenogenesis. First one, complete parthenogenesis. So complete parthenogenesis is the phenomenon where an organism produces only the parthenogenesis. There is no biparental sexual reproduction. Usually, the individuals have only females, no males are seen. It is also called as a parthenogenesis. Examples: Bedelloid rotifers. So complete parthenogenesis is the phenomenon where an organism reproduced only by the parthenogenesis. There is no biparental sexual reproductions. Only for single types of sexual parent occurs in female. No males are seen in types of reproductions. So examples of rotifer species. Next one, incomplete parthenogenesis. Incomplete parthenogenesis. Incomplete parthenogenesis is the phenomenon where the organism reproduces both by sexual reproduction and parthenogenesis. Incomplete. So partially the reproduction occurs in sexual method, then partially occurs in parthenogenesis. It is also called as partial parthenogenesis. Examples of honeybees: the fertilized eggs developed into a female and the unfertilized eggs developed into a males. The bees or beehives contain three types of bees, is namely queen bee, worker bee, and male bee or drone bees. The queen bees and worker bees is female. Drone bees only for male. The female bees are developed from fertilized eggs. The male bees are developed in unfertilized eggs. This is a queen bee. The queen bee form or laying the eggs. Some of the eggs is fertilized in diploid condition. Some of the eggs is unfertilized. It is a haploid condition. The diploid condition eggs or fertilized eggs to form a female offsprings. Then unfertilized eggs or haploid conditions to form a male offspring. The female offspring is a queen bee, diploid in nature, and worker bee, diploid in nature, and drone bee, haploid in nature. So it is a partial parthenogenesis or incomplete parthenogenesis. Next one, pedogenetic parthenogenesis. The parthenogenesis that occurs in the larva is called as pedogenesis. Parthenogenesis occurs in the larval stages. The larva produce another larva. The larva produce another larva. Does not undergo a sexual reproduction. These types of reproductions is called pedogenetic parthenogenesis. So the parthenogenesis that occurs in the larva is called as pedogenesis. In pedogenesis, the larva undergoes a parthenogenesis and form a new larva. Examples: sporocyst and radial view of a liver fluke. Radial liver flukes. It is the examples of pedogenetic parthenogenesis. Next one, artificial parthenogenesis. Artificially, the parthenogenesis occurs in natural process. It's called a natural parthenogenesis. The parthenogenesis is stimulated by the artificial process is called as artificial parthenogenesis. The process by which an unfertilized eggs induced to develop into an embryo by physical or chemical stimuli is called as artificial parthenogenesis. Physically or chemically, the unfertilized eggs stimulate into form or produce a new individuals is called as artificial parthenogenesis. So factors like temperatures, UV rays and certain chemicals like chloroform and induce them parthenogenesis. Such factors is called parthenogenetic agent. Physical and chemical factors, like physical factors, temperature, UV rays and chemical factors, chloroform. It is a parthenogenetic agent. So see the picture. It is a fish. It's laying of eggs. So check the radiation and eggs converted into diploid. The diploid eggs furtherly developed to form into a new individuals. This is the artificial parthenogenesis. Next one, classification of animals based on the sites of embryonic developments. The embryo is developed in a different sites, outside of the body and inside of the body. It's depending upon the animals are classified in three types, namely oviparous, viviparous and oviviviparous. First one, oviparous animal. Oviparous animals are the egg layers. Egg layers. The embryonic development occurs in the inside the laying egg and not in the mother's body. Embryo is nourished by the yolk in the eggs. The oviparous animals, embryo is developed the outside of the mother body. Outside of the mother body. Examples of egg laying animals. So all the egg laying animals, the embryo is developed the outside of the body. Next one, viviparous. In viviparous animal, the embryonic development occurs inside the mother body and finally delivered as live young one. Embryo is nourished by the mothers through the placenta. So it is a major difference for viviparous and oviparous. It is also a important questions. Mention the difference between the viviparous animals and oviparous animals. So viviparous animals, the embryo is developed inside of the mother body. Oviparous animals, the embryo is developed the outside of the body. So nourishments through the yolk. So viviparous animals nourishment is supplied through the placenta. Next one, oviviviparous animal. In oviviviparous, the embryo developed inside the egg remain the mother's body until they are ready to hatch. Embryo has no placental connection with mother but get nourished from yolk sac. So it is them special types of animal, oviviviparous. The ovum or eggs develop the inside of the mother body or parental body. The young ones develop to the inside of the egg. Inside of the eggs. So, but the young one does not get the nutrients from the mother through the placenta. This types of animals is called oviviviparous animals. Example, shark.